And as we're talking about trying to engineer a transcription factor, I brought up two different examples where you're trying to do something that's more general, maybe be able to activate more sequences, maybe have it be applicable in different kinds of organisms, trying to bind also a broader range of small molecules. Well, we can also take a step back and say, beyond those things, what kind of property do we want from our transcription factor? And thinking from an electrical engineering and programming kind of perspective, I want to bring up this idea of analog versus digital response. And I want to ask the audience, which response do you think is better? Which one do you want? Response on the left, response on the right? Which one, if you might have any intuition here, would you think is referred to as analog versus digital, and why? So I'll pause for some answers.
I think the left will be analog and the right would be digital, because digital is like 1 versus 0, on or off.
And that's right. So keep going. So which one do you want?
Well, I don't think it's fair to say which one do you want, because it's all context dependent. Like if I just want to know how much of someone's moving furniture, I'm sorry. If I want to know how much of my substrate I have, I want analog because it kind of goes linearly. But if I just want to know if it's there or if it's not, then I want to use digital.
That's perfect. That's an excellent response. Cool. Yeah, so that's right. That right on what's analog versus digital, as well as at least one reason why you might want one or the other, and the more general answer of context dependent. You can think of other reasons why you might want one or the other. So good job there. So let's talk about what actually controls the shape. I mean, do you actually need, if you really wanted one and you have the other, but you have the right small molecule bound and you've got your operator sites like all set up, how can you go from one to the other? And so that's this general phenomenon that leads to this kind of behavior in the first place, is cooperativity. Trying to bring some more math into this course, although it seems like plenty of it is starting to emerge in your homework. There's an equation known as the Hill equation. I think sometimes it's called the Hill-Langmuir equation. You've got terms here that have to do with familiar things that you've seen in kinetics, ligand concentrations. So this isn't an enzyme in this case. So you've got your ligand, you've got a dissociation constant, and you can see this plotted here. And maybe some of you, maybe several of you have seen this before. This is the idea of, as you have an increasing ligand concentration here, shown on a log scale, you can get the fraction of your receptor that's bound by ligand can suddenly increase, or it can remain relatively analog or linear in response, as described before. Can any of you think of a system in human biology that's cooperative? Or is this an example that would be familiar to you? Say hemoglobin. So maybe that question is ill-posed, but have you guys come across like how hemoglobin is cooperative, and or things like carbon monoxide poisoning? Is that worth me explaining?
Yeah, so we went over it in like pretty strong detail in biochemistry, but I kind of forget the exact details. So if you want to go over it quickly, I think it'd be good.
Okay, I'll do that. I'm recognizing also that it's noon, so I'll try to wrap up soon. But yeah, in general, hemoglobin is one of the ways that we bind oxygen, and it's cooperative. Once you have, I think it has 4 binding sites, and once you have 1 oxygen bound, you get a greater affinity essentially for your other oxygens. And one of the dangers with carbon monoxide poisoning is it finds the same binding sites, and it's also cooperative. So it is very possible to immediately, from a very small concentration, get really tight binding, and then essentially have no ability to bind oxygen. That's one of those examples that I've come across from a long time ago that I didn't bother to double check right before this lecture. So I hope I explained it correctly. Perhaps a different or better example of cooperativity when it was first studied and emerged in the literature was in bacteriophage. So taking a step back, bacteriophage are viruses that infect bacteria. And so there's a bacteriophage called Lambda, where a lot of classical studies were done, and it infects E. coli. Lambda is interesting because it's a lytic bacteria. It will eventually cause cells to burst, releasing its viral contents everywhere. But it has to not do that until a certain time. It has to go into cells and make enough of itself to then be able to once it's built up a large enough concentration, enter this lytic phase. And so Lambda actually, it was known a long time ago that Lambda has cooperative binding. It binds, it has a repressor, the Lambda repressor. It binds cooperatively to multiple sites across its genome that activates its own expression, and it represses the genes that are required for cell lysis. And so, you've got these repressors that, like we saw with those operator sites, you've got the binding sites, but then once one is bound to a binding site, it's much easier for the other to come in and bind. And the two help stabilize each other, such that they don't unbind as quickly. If you imagine the on-off rate of one, once you've got another, if it wants to let go, it's still kind of held on. So this is all just to say mechanistically what is the cause of cooperativity. Hopefully that explanation made some sense. Interestingly, in prokaryotes, you see a limited amount of transcription factor cooperativity, relatively speaking. Of course, in the example that I just gave you with VNR, there were 2 sites, and you do see a lot of dimer operator kind of sites. But actually in eukaryotic transcription, cooperativity is really critical, or at least thought to be so. So anyway, this value here, n, that's known as the Hill coefficient, and it's a parameter that you'll be able to play with in your homework, thus getting a better sense for cooperativity. In terms of how you might, from an engineering standpoint, change cooperativity, based on everything that I just mentioned, you can imagine that there's an interface or a region between 2, say, dimers that bind cooperatively, and that you could potentially eliminate or modify the surface residues that create that favorable binding, such that these 2 identical things could bind entirely independently. Or you could imagine that you create more associations, maybe even very permanent associations, such that now you've essentially made it as cooperative as it could be. Like it's really just one unit, but there's still 2 sites, so it moves together. And so these are, from an engineering structural standpoint, how you can manipulate cooperativity. So I'll just cover a couple more slides and then wrap up today. Thinking about applications for biosensors, the very common one that I was just describing also with Banalate was just monitoring. And so this example nicely illustrates with a metabolic pathway how you can get some product here represented with a square, and how your triangle, circle, or hexagon precursors don't bind your transcription factor, so you don't get fluorescent protein, but then when you have the product, you get fluorescence, and hopefully proportional to your product. Or maybe you only want it to be binary, but in a metabolic engineering context, you probably want it to be proportional if it's your product. because if it is proportional, say, related to your product, then it's easier to imagine directed evolution of either your organism or the critical enzyme that, or any of the enzymes in the pathway, but probably a critical one if there is a rate limiting one. So I think we've talked about this when we talked about rational protein engineering, and then the lecture that I added on to the syllabus about directed evolution. But hopefully now these pieces are coming together a little bit more clearly. How you can, and we still haven't actually talked, that'll be next time, how you can perform mutagenesis to generate diversity in the genome. When you have either in the genome or on a plasmid, I should say, when you have this diversity, you have it in a library. And then, for example, this shows sort of a microfluidic and self-sorting approach, such that things that are more fluorescent might go down one channel versus another. And ultimately, then you can collect things that are more fluorescent, which probably have a better enzyme, and iterate in the next round of this circle. And this is kind of shown as if done continuously, but it doesn't, it can be done continually